So before I begin, are there any questions? Will stuff in section meaning what? I haven't thought about that. You don't have your hard copy from section?
No, I lost it.
Lost it.
So maybe in that case, ask the GSI for a copy and send them an email. That's the best solution.
Okay. Okay, so last lecture, Elliot gave you sort of an overview of sort of the astronomical world, and my goal in this lecture is to give you an overview of sort of the biological evolutionary world. Both of these are very daunting tasks, as both disciplines are huge, and a 75-minute lecture, 80-minute lecture is tough. So two of the themes that from Elliot's lecture that are going to transfer over to this lecture are, first of all, Elliot emphasized the extreme importance of the unseen in changing our worldview. So once Galileo invented the telescope, the fact that he could see moons orbiting around Jupiter or the phases of Venus utterly destroyed, finally, the Earth-centric view of the universe. And so the ability to see things that can't be seen with the naked eye, transformed our understanding. And so it is also true in biology that our ability to see things without our naked eye have completely changed our view of our place in the living universe. So that's the first theme. The second theme is just the incredible scale of things, astronomical. In the case of biology, most of the scale is really, really tiny, but in some cases there are also some very large numbers. So keep in mind the scale of things and also sorry, my brain just fizzed. The issue of the seen versus the unseen in our understanding of how we are placed in the universe. Finally, the last theme was that in astronomy we often get a lot of information by using things other than visible light, and we'll find exactly the same thing also happens in biology. So in this lecture, I'm going to do three things. The first is I'm going to look at the 6 major groups of life that are currently living on Earth. There are possibly other groups that are now extinct. We'll look at that later in the course. I will then look at the evidence that all life on Earth shares a common ancestor. So why do we believe that? And so I'll give you the essence of those arguments. And then finally, I'll finish up with a universal measuring stick for life. That is, can we put all of life on sort of one set of axes? And when we do that, we will discover 2 quite remarkable things about ourselves and the evolution of life. So I'm going to start with the 6 major groups of life. So the first 3 groups are the animals, fungi, and plants. They are all multicellular, and for the most part are middle-sized, ranging to huge. These are the major groups that Linnaeus recognized, and these are groups that are easily accessible by the naked eye. And so they've been known for several centuries and classified for several centuries as well. Formally, they're called kingdoms, but that's really not a big deal for this course. So, get your colored sheets of paper out. First question. How many times do you think multicellularity actually evolved? So minimally 3, animals, plants, and fungi. And as we'll see, they each have independent origins of multicellularity. So the last common ancestor of those 3 things was not multicellular. And so the question is, have a chat amongst yourselves. What do you think the number is? 3 times, 6 times, 12, 24. Okay, have a shot. Hard to know without evidence. Some 24s. Hold up your sheets high. Some 6s, some 12s. Any whites? Some whites. Okay. So it turns out that multicellularity has arisen in the order of 24 times that we are aware of in the history of life, which is an awful lot more than 3. Now, you'll see another 4 of those as this lecture proceeds. But one of the questions, which is an open question, actually we've just submitted a paper discussing this to a journal, a colleague and I, is of all the 24, there are only 3 that have produced a huge number of species and a great range of morphological complexity. Morphology just referring to the physical form of the organism. And so the question is, why is it just 3 or so that have produced most of the disparity, most of the differences we see in the biological kingdom? So that's one of the questions of origins we'll be dealing with. The other 3 groups are shown here. Now, in the reading, these are also called kingdoms. This is not a generally accepted word, and so I've just replaced up the top here groups, so don't worry too much about the terminology, but they're not formally kingdoms. The 3 groups are bacteria, and then another group of bacteria called archaea, and then the so-called protists. And what is significant about these 3 groups is that they were not recognized by Linnaeus. And the reason why they were not recognized is because they are largely invisible. So we get the unseen striking us immediately. We can't see bacteria or archaebacteria with the naked eye unless they form huge colonies. We can't see most protists, but we can see some, as you will see a little bit later. How many of you heard of Giardia? That you can collect if you drink water up in the Sierra. So Giardia is one of these protists, for example. Okay. So now I just want to give a very, very brief overview of these 6 kingdoms. So the kingdom 1 is the animals. Interestingly, the origins of animals are in the ocean, and it only took sometimes later that we invaded land, and we'll be dealing with that origin also later in the course. But it is interesting that the legacy of our oceanic origin is still with us. The salt concentration in our blood is the same as seawater. Could be a coincidence? Probably not. Why not? Any ideas as to why salt blood needs to have the same salt concentration as seawater if you live in the ocean? Yeah, so have you heard of osmosis? So if you've got a high salt concentration and a low salt concentration, the high salt concentration sucks the water out of the low concentration. And so it looks like that our blood is basically isoosmotic with seawater. And we've retained that heritage in our blood even though we're no longer in contact with water in that way. There are about 30 fundamental body plans amongst the animals. So vertebrates and their relatives constitute one, the arthropods, the insects is another, sea urchins and starfish another. There are a whole bunch of wormy ones. They originate suddenly and dramatically in the Cambrian explosion about 500 million years ago, and that we will also look at in another lecture. Actually, not next week, but the week after. And as you are probably aware, there are spectacular morphologies and behaviors associated with the animals. So that's the synoptic overview of animals, nothing more to know. The second kingdom are the plants. They exhibit a great range of morphologies, though not as diverse as animals, and can reach absolutely colossal sizes. Plants arrived on land after animals evolved in the ocean, and for a long time there were no plants on land. So imagine a world with nothing this sort of size in it. So prior to the evolution of land plants, the world was a fundamentally different place, both in terms of organisms but also atmospheric chemistry, and we'll be looking at that also in a later lecture. Plants photosynthesize with their chloroplasts, and there are many other groups that have chloroplasts that are not plants. So plants have chloroplasts to harness harness energy from sunlight, but there are many other groups of organisms that have chloroplasts that have nothing to do with plants in the sense of being large, woody, flowery things. And plants typically extract nutrient and water from the soil with roots. But the first plants on land didn't have roots. Okay. The third one are the fungi, mushrooms, etc. They live on decayed organic remains. They are more closely related to animals than to plants, And one of the ways we know that is that fungi are fairly stiff in the way that insects are stiff, and so they have a substance called chitin that helps give the stiffness of their bodies. And that's one of the pieces of evidence that fungi and animals are closely related to each other. We'll see a little bit later in the lecture the real reason for understanding that relationship. Okay, so those are the three large groups that Linnaeus recognized that dominate our visual world. This is really just for gratuitous fun. Each group has produced phenomenally large organisms. The largest animal that has ever lived, including the dinosaurs, is the blue whale at about 190 tons, which is pretty close to a North American tonnies. The largest land plant, the giant sequoia in terms of mass. How many of you have seen General Sherman? Yeah, it is truly spectacular. I'd take an opportunity if you go to the Sierras to see that. It is awe-inspiring. But there's also some quaking aspen where all of these individual trees are connected underground by the same root system and therefore are actually the same organism, even though it looks like it is multiple individual trees. And so when you estimate the mass of that single organism, it turns out it is a lot bigger than General Sherman at about 6,000 tons, covers 100 acres, that's about 100 football fields, and is in the order of 80,000 years old.
How can you tell that they're all connected by the same root system?
I think they just started to dig. It's a good question, actually. I don't know, because that's a lot of area to dig without destroying it. I also don't know whether they did DNA on it or not and saw that they were identically the same, which is the way I would go about it. And then fungi also have huge underground networks, and then they pop their little heads up individually. And so here's a mushroom here in eastern Oregon that looks like it's about 605 tons, covering about 2,200 acres. So the subterranean world is also incredibly significant for both plants and fungi. Okay. So then we've looked briefly at plants, fungi, and animals. And so these are all eukaryotes. So we have a big split in the animal worlds between eukaryotes and prokaryotes. And now what I'm going to do is briefly review that distinction for you, because it, again, will be important a little bit later in the lecture. So the fundamental building block of life is the cell. Some people tend to think of it as being the DNA or the RNA. It's really the cell that's in charge, and it manipulates the DNA in order to grow and feed and reproduce, communicate with other cells, and there's a whole lot of other structures. And so the eukaryotic cell is shown here, which is much more complicated than the prokaryotic cell, which belongs to bacteria. So these things are basically in the world of the invisible. This is the world of the mostly visible. So in terms of size, eukaryotic cells are on average 1 or 2 orders of magnitude larger than prokaryotic cells, which are larger than viruses, which are larger than proteins, which are larger than small molecules like oxygen, which are larger than atoms, which is hardly a surprise given that they're made of atoms. And so what we've done here is added in the wavelengths of light on the scale here. And so you can see this is the scale length here. Notice visible light here is at about in the middle of the prokaryotic cell size. It's pretty hard to see things with visible light if things are smaller than visible light, right? And so we really can only visualize directly the smaller prokaryotic cells and viruses and proteins if we use a different type of light, like ultraviolet running all the way down into X-rays. And so, as in astronomy, where we look at the world with different types of light, so in biology, we also look at the small part of the living world with different types of light, ultraviolet and X-rays. Now, how many of you have seen this? Oops, sorry. Yeah. Well, how many of you have seen? So, an exception. So here, the largest eukaryotic cells peak out at about 1 millimeter, but there are cells that are vastly larger than that. Right? An egg is a single cell. right? So this one should go up to the order of centimeters to meters. So sometimes eukaryotic cells can get really, really huge. This is an egg of the elephant bird that's recently went extinct in Madagascar. So larger than an ostrich or an emu or a moa, if you know the extinct New Zealand bird. Okay, so how many of you, if you look up on the Berkeley Hills, have seen this? So when you go outside, yeah. So anybody know what it is? Do you know what it is? So what it is, yeah, so this is something called the advanced light source, and what it does is it produces ultraviolet and X-rays for high-resolution imaging. And it's one of the nation's premier facilities for using wavelengths in this range to look at the tiny biological world. So people from San Francisco State, UCSF use it to visualize DNA, protein. So have a look next time you're there. And it looks a little bit drab from the outside. It's pretty high-tech on the inside. And actually, we paleontologists use it too. We can get down to 0.25 nanometers, and with the X-rays you can see inside rock. So we can visualize cellular-level structures inside fossils by using this. So it's got a huge range. I think there's 42 beamlines with different wavelengths for different purposes. So that's what that thing is up on the hill here. And it's one of the things that makes Berkeley truly exceptional. It's not just what's on campus, it's what's around campus. So eukaryotes have a membrane-enclosed nucleus, shown here, and that houses our DNA. And so they're in the order of 10 to 90,000 genes. We have roughly 20,000 genes in there. They also have inside them mitochondria. How many of you have heard of mitochondria? Yeah, okay, good. So we have mitochondria also sitting inside the eukaryotic cell. The mitochondria produce the cell's energy, ATP, and they're a little bit unusual in that they have their own circular DNA. Now, it's very tiny. It has just 35 genes, which is an awful lot less than 35,000 genes for larger animals. The other thing that's a little bit unusual about it, it has two membranes around it, just like the nucleus does. Cells have one membrane. The mitochondria has two membranes. And that's going to be important a little bit later. Mitochondria are typically shown as isolated. How many of you have seen textbook pictures like this in high school? That's the way we all got taught, and I knocked on the doors of one of my colleagues. Yes, question? They are only in eukaryotes. They are only in eukaryotes. Good question. So mitochondria are typically shown as isolated entities. They're sort of viewed as the little sort of power plants of the cells sitting there like a nuclear or gas or coal power plant. Turns out it is completely untrue. And so this is a video taken by George Brooks in the Department of Integrative Biology, my department. And so the mitochondria are about this sort of size here, and they form rivers inside your cell that move and change as the cell's needs change. So the cell itself is not static. It has a cytoskeleton that moves, and there are typically tens to thousands of mitochondria that move around inside the cell, forming these rivers. Now, if you take a cross-section through a cell, cut it, then you will catch one of these streams, and that's what it looked like. And so I think the error is, because most of the time when you slice a cell to look at under a scanning electron microscope, you just cut one of those streams like cutting a wire, and you just see the end of it, the cross-section, and you don't realize that you're seeing part of a pipe or a stream of mitochondria. So let me, yeah.
That's one cell?
One cell is, I've forgotten where the boundaries are. It's in the order of one cell. Question?
What exactly is the boundary between cells? Like when does one cell end and
Most of the time, cells have very distinct cell walls or cell membranes. So they're dead easy to see. It's like lots of little water-filled balloons stuck next to each other. And there are connections between them. There's a very clear membrane. Now, plants, in addition to having the nucleus and mitochondria, also have chloroplasts. The chloroplasts perform photosynthesis, harnessing the energy from the sun. They also have their own circular DNA with typically 100 genes in them. And they also have two membranes. So just like the mitochondria, they've got their own DNA, not many genes, and two membranes. And so that will also be important a little bit later in the lecture. So then, returning to this diagram then, plants, fungi, and animals are eukaryotes with large cells, with nuclei, mitochondria, and in the case of plants, chloroplasts. So the fourth group is the group called protists, and this is a really ugly group of organisms. Most biologists don't use the term, and you'll see why. And the reason why it's an ugly group of organisms is their definition is eukaryotes that are not animals, plants, or fungi. So they're every other eukaryote that happens not to be one of those three groups. And that's a very frustrating and irritating definition. But it captures a part of the biological world, and so for the purposes of this course, we're going to use it. Some have skeletons. Some have many nuclei per cell, thousands sometimes. Some of them, most of them, are single cells. Some of them are multicellular. Now, I don't mean animals, plants, and fungi, but some of the protists themselves are multicellular. Interestingly, some of them are both multicellular and single-celled. And I'll show you that in just a moment. But our biology as animals is incredibly rigid. Two sexes, fixed number of nuclei in the cell. When you get into the world of the protists, all the rules break down. There are things with multiple sexes, things that change sex, multiple nuclei, incredible life history strategies. Several groups are multicellular, and so the three most famous are the brown algae, of which I think the kelp is the most spectacular. So obviously that's an extraordinarily large organism that's not plant, not fungi, not animal. So this is our fourth independent evolution of multicellularity. There are the red algae. There are the golden algae. So these are protists. So what biologists do is erect each of these into their own kingdoms. And then it starts to get a little bit out of control. Question?
Why don't algae protists plants?
Because they have a completely separate evolutionary origin. That's really the reason for it. And we'll see that a little bit later in the lecture. And then slime molds are really spectacular. Slime molds are basically an amoeba, which is a blobby thing that crawls across the ground. And what happens when they want to reproduce is the individual amoebal cells gather together, they swarm together and start to grow a stalk that then grows up and produces a reproductive organ that then produces spores. So it spends most of its life as separate individual organisms, and then when it's time to reproduce, they aggregate into a multicellular structure, which then produce reproductive spores. So it's both single-celled and multi-celled. So again, for us, we're multicellular. There's nothing we can do about it. We're incredibly inflexible that way. Not these guys. So a very, very interesting range of biologies that these different groups can exhibit. Question, yeah.
Would all the amoeba be identical when they're separated, or?
No, they're not. And so first of all, let me make one point. There are many types of amoeba. This is just one of them. And so it produces a really interesting dilemma, is given that they're all different from each other, how do they cooperate in such a way, given that only a subset will produce spores? So that produces an incredibly interesting question for the evolution of cooperative behavior. There's one there. Yeah.
This might sound weird initially, but before they become the slime mold and multicellular stage, did the single cell amoebas are they like stem cells, and then when they come together they start switching to the protein?
Oh, that's an uneducated question. That's terrific. Yeah. can you view them as stem cells in the sense that each one of them has the potential to become any one of the different parts of the structure? And I think in some sense the answer is yes, they are like stem cells. And so a lot of people actually look at them in that context because they cross that boundary freely backwards and forwards between being a stem cell and not a stem cell. Yeah, first of all, there's no such thing as an uneducated question, anytime, anywhere, so feel free. The question there and then there, yeah.
After becoming a multicellular organism and producing its spores, do they break back down into being single cells, or do they just die after?
I think they go back to being single cells again. I'm trying to remember video I've seen of it. I think it just collapses into a blob again and disarticulates, but I don't know for sure. If you just put slime mold into YouTube, you'll get video of this. It's kind of fun to do. I suggest that. Okay. One more, yeah. What they do is they ingest small particles like bacteria. So there's a little bacterium there, and that's going to become important later too. So they feed on prokaryotes and other small protists. One last, yeah. Yes. Well, I don't know whether they go through multiple reproductive phases or not. So, obviously many animals do, but almost every octopus species in the world dies immediately after reproduction. So the number of species that they reproduce once and that's it, they're gone. We're not one of those species, for example. And so I don't know what these guys do afterwards. So there's lots of variation in that as well. Okay, group 5, and now we're going to drop into the prokaryotic world. So far, everything has been a eukaryote with a nucleus, a big cell, mitochondria. The bacteria, they used to be called eubacteria, as in true bacteria, and now they've just become bacteria again. They are prokaryotes. They are typically very small with very little morphology. They're mostly little blobs. We saw pictures of those on the tip of the pin in my first lecture. They have a huge range of life habitats. They can live almost anywhere. Examples include E. coli that lives in your gut. One of the interesting questions is, where was E. coli living before animals had evolved? Because there weren't any guts. Open question, we don't know. And things like cyanobacteria, which also photosynthesize. And so here is a multicellular cyanobacterium. So this thing has multiple cells. And what I'd like you to note very carefully is that this cell here are different shapes and sizes from the rest of them. So if you starve this guy of nitrogen, one of its cells transforms into this cell. It turns on about 1,000 genes, and then it fixes nitrogen directly. And so this is not only multicellular, but it has more than one cell type some of the time. So that's now the 7th example of multicellularity that we've looked at in this lecture. And critically important is that it also photosynthesizes, even though it's a bacterium. So plants in that way are not special at all. Yeah. Yes. Multicellular eukaryotes do exist. Some of them have spectacular colony forms, for example. Multicellular prokaryotes do exist. Some of them have spectacular colony forms. Yeah. We tend to think of ourselves as being highly privileged in our own biologies. We turn out not to be so privileged. The same story we saw in Eliot's lecture, where we thought we were privileged being the center of the universe, and it turns out we're not nearly as special in that sort of way biologically. Yeah. Is it? Well, what's the difference? Yeah, there is a difference in the sense that what we do, of course, is we use our cells to make higher order structures, which we call organs, like a heart or a liver or a kidney or bone, where this thing isn't making higher order structures. It's still just making a chain. So you might view that as a colony. It's still multicellular, but it's very different from our type of multicellularity. And so we'll look at that a little bit in a later lecture. What is it that makes our genes and cells capable of producing higher level structures rather than relatively simple multicellular structures. So actually it's a very deep question and difficult to answer. So then bacteria are actually abundant. How many of you have been to this place? Yeah, it's really, really nice. The Grand Prismatic Spring in Yellowstone, Wyoming. All of these colors here are different types of bacteria living in different acidities and temperatures of water. The last group are the archaea, or archaebacteria. Again, the names keep on shifting a little bit. They are prokaryotes, and they look like bacteria. So under the appropriate light, they look bacteria. They have unique cell membranes, however, which distinguishes them from bacteria. They have a larger range of habitats than bacteria. They can derive their energy from sugars, like pretty much everybody else. Plants take sunlight, turn CO2 into sugar, and then use that sugar. Well, we eat it in the forms of fruit and vegetables. We eat things and turn it into sugar and then convert it into a whole bunch of other different things. Bacteria do the same. Archaebacteria can also get energy from metals and even hydrogen gas. So archaebacteria can do a whole bunch of things that eukaryotes and bacteria don't do in terms of getting energy out of the environment. Some of them don't need sunlight or sugars as the primary way. They don't need CO2, carbon, as the primary way of making their bodies. So the study of archaebacteria has become critically important to understanding both the origin of life, but also whether or not life exists elsewhere in the universe, because it means there's a wider range of environments that can support life than we understood before we discovered archaebacteria. So really, really important organisms, as tiny and as dull as they look like from the outside. And we'll see shortly why they're separated from the bacteria. And so archaebacteria, how many flying into SFO have seen these red ponds across the San Francisco Bay? So those are archaebacteria. They are called halobacteria. Halo simply means salt. So these guys live off salt. And so sometimes they can be enormously extensive. Many of them live in extreme environments like black smokers that live in the abyssal depths on the seafloor, where there's geologic gases coming up through the sea crust, and they live at enormously hot temperatures, above boiling temperature, if the temperature was at the surface. So incredible biologies. Okay, now a point of clarification here. This bubble diagram sort of captures the eukaryotic, prokaryotic structure and the 6 groups, but it's not an evolutionary tree. This is the evolutionary tree, the relatedness of the groups superimposed on this diagram. We're going to deal with evolutionary trees later in the course. You're going to have a section on building them. But I'd just like you to note that, first of all, there's a last universal common ancestor, and I'll show you why we believe this in the next section of the lecture. Archaebacteria and eukaryotes are more closely related to each other than either is to eubacteria. So our nearest relatives as eukaryotes are the archaebacteria. Question? I'm using them interchangeably. And so this diagram I like, but it's irritating because it has old terminology there. An archaebacteria here. I'm not going to trick you up on that at all in this course. Question?
These are various forms of life. What makes them life?
What makes them life? That's a really good question. Let me get to that in just a moment. I'm going to cover that in the Origin of Life lecture, but I've got a slide coming up where I'll try to address that. Yeah, question? So if time is going up this way, there's the last common ancestor, which spawns off the bacteria, and then evolution proceeds a little bit further, and then it spawns off the archaebacteria and the eukaryotes after the bacteria have come out. Now, we'll deal with this in more detail. The second one, which is very interesting, is that the plants, the fungi, and the animals each have their origins in unicellular protists. So there's a protist, that is a eukaryote, that was single-celled. It then became multi-celled, and that multi-celled thing became a plant. A completely different unicell protist evolved into a multi-celled protist, and that turned into a fungi. A third completely different unicell protist evolved into a multicell protist, which became animals. That one is called choanoflagellates, and they still exist with us today. So each of these multicellular groups evolved from a separate single-celled protist that went through a multicellular protist stage. And the last one, which I've highlighted before, is animals and fungi are more closely related to each other than either is to plants. Question and then question.
How do we know anything about whether life could have originated in several times?
That's the next part of the lecture. We'll deal with that. It's a very good question. How do we know that life has a last universal common ancestor? Yeah. Well, so then in here there's another one that's multicellular. So this diagram basically is referring to multicellular protists. So it's a little bit irritating because this diagram is still sort of animal-centric. So it should have multicellular lines coming off in several different places. Because it's not a tree, it's just capturing the 6 major groups. Okay, we'll deal with this in more detail later, but I just want to clarify that because last time when I presented it, people started to get confused because they started to interpret the diagram as a tree, and it's not. Okay, what about viruses? So another question is, what is it that defines life? So let's deal with viruses. Virus, like a computer virus, viruses are tiny bits of packaged code, DNA and RNA, that are often packaged in some way that are injected, in this case, or get engulfed in some way so that their DNA or RNA gets incorporated into the host DNA. So the virus DNA or RNA gets inserted into the animal or plant or fungi's or bacteria's DNA. And then what it does is it uses that organism's cellular machinery to reproduce. Like a computer code. The code inserts itself into your computer, and then your computer reproduces it with the machinery that it has. So opinions differ on whether viruses are a form or life of not. They're information-rich. They reproduce, but they can't reproduce by themselves. So one of the criteria is an organism is something that can reproduce by itself. Also, these guys don't have any metabolism. They can't do anything. They're just a piece of information. If you think of a computer, it has metabolism. There's a hard drive, there's a screen, there's memory. It has lots of processes. And so organisms all have processes to grow, to reproduce, to repair their cells, to repel invaders like viruses. Viruses have none of that. They're just little bits of information that insert into the cell and then use that cell's machinery to produce more copies of themselves. So that's as far as I'm going to get to a definition of life at this point. So that's how viruses fit in, which is we don't really know how they fit in. Most people view them as being not life, but being on the edge of life. And their origin is unknown. Do they predate the existence of life? Like a computer virus, could they only come into existence once life came into existence? Computer viruses didn't exist until there was computers and a way of communicating between the computers. So in some respects, the computer virus is a really good analogy for a virus. So, question and the question. So, good question. There are a lot of the viruses, like AIDS, for example, does not actually get its DNA into our cells. So it actually just reproduces in our bodies. So in that sense, the fever is the immune response to the virus. So there are other ways, of having them reproduce. So thanks for asking that question. As usual, biology is way more complicated than a simple description.
So was mitochondria?
We don't think so. We think we know where mitochondria come from, and we'll get to that at the end of the lecture. So that's a good question. Right. Okay, so now, what is the evidence that all things actually share a common ancestor? Which is what I just asserted in a slide or two ago. The best evidence comes from shared complex features, including Use of DNA and RNA, the same amino acids, the same genetic code, and the same machinery. I'm going to review these quickly. But why do I emphasize complex features? Why is that an essential component of the argument? Yeah, right. So the converse way of saying it, shared simple features don't guarantee anything. My name is Charles. Maybe there's another Charles out there. Are we closely related to each other? No. It's a common name. Organisms are made of atoms. Wow, that means they're related to each other. That's daft. Right. Really complicated things are hard to evolve independently. So if you have the same complicated thing, it suggests that they have the same origin, single origin. Okay, so let's look at that. So first of all organisms use DNA and RNA to code for information for making proteins and the RNA itself. So here's a picture of the DNA molecule. Each little sphere is an atom. So this is on the scale length of an atom. RNA uses a different base we're not going to worry about for this lecture. The size is tiny. So each of these little rungs in this ladder here is called a base pair, and it's in the order of 10 to the minus 9, a billionth of a meter. So it's really, really tiny. So this is the famed double helix. So everybody uses that. So how much DNA do you have? Turns out we have 3 gigabases, 3 billion base pairs. So 3 billion of these. 3 billion of those individual base pairs. Question?
Do all organisms have the double helix DNA type?
There are some viruses that have single-stranded rather than double-stranded. But as far as I know, all bacteria, archaea, protists, animals, plants, and fungi have double-stranded DNA. So the question is, so that's how much DNA you have, 3 billion. So now we're getting into astronomical sizes again in terms of numbers. Question?
Is there some sort of advantage for the virus having a single cell or a single strand DNA rather than the double helix?
I'm going to answer that in a funny way, which is it exists, so the answer is yes. I don't know the molecular mechanism by which it reproduces where the single strand operates better. So there are many ways that DNA and RNA are manipulated by cells, and some of them can manipulate single-stranded RNA. In fact, when a gene gets converted into a protein in your body, it goes through a single-stranded RNA intermediate. So that's a common part of the life cycle. So it codes for about 20,000 genes, which is only 2% of your genome. So 98% of your genome does not actually code for genes, which itself is an incredible mystery, and I'm not going to deal with it here. So question, if you were to unwind the DNA from one cell, how long would it be? 2 millionths of a meter, 2 thousandths of a meter, 2 hundredths of a meter, 2 meters. Okay, so we're getting a whole bunch of reds quickly, which suggests that there's some reading been done. Excellent. Yeah. So that's interesting. You know, even though DNA is at the level of atoms, you have so much of it that if you took all of it, ran 2 meters long, which is really quite incredible. Question.
This also feels like it's like a silly question, but I'm just not quite sure. How does DNA and the genome relate? Like, what exactly?
The genome is made of DNA. The genome is made of DNA.
So all the DNA is just called the genome?
Yes, good question. All the DNA is called the genome. Yeah, okay, that's things that it doesn't occur to you aren't clear, so I'll change that for next year. Yeah. Now, what's amazing is if you type out the DNA from one cell, ATGC, ATGC, GCTC, it would fill about 2,000 volumes the size of Tolkien's Lord of the Rings, right? Which is about 1,000 pages long, the version I read when I was a kid. So that 2 meters of DNA is an incredible number of letters. So how much DNA do we actually have? We have about 10 to the 14 or more cells. So again, this is hugely astronomical numbers. So then the question is, if you took all the DNA from all your cells, how far would it extend? San Francisco to LA? San Francisco to Australia? I have no idea why I picked that continent. Pluto and back, the nearest galaxy. What do you think? I like that, all four colors. Let's see, some green, some blue, some red. Typed out, all the DNA from all your cells would fill about, I can't do that, 100 billion 500 gigabyte hard drives. Right? Now, it's the same DNA over and over and over again, but that's how much is there. And so I say with cheek, and we thought we invented the information age. We are not even close. Not even close to what organisms do. So it's no wonder that we can't produce artificial intelligence yet, for example, that matches our own minds or brains. We are not even close to what biology does. So that's where these astronomical numbers and the combinatorial potential of that vastly exceeds our feeble technological advances. So that's really important to understand. Okay, next thing is we all use the same amino acids. So this is a molecule. An amino acid has a nitrogen at one end and a CO2 at the other end, and then a complex carbon chain here. It's this R that dictates the identity of the amino acid. Don't worry about the details. There are about 500 types of amino acid known. All life uses just 20, the same 20. If life had evolved independently separate times, you'd expect a different set of amino acids if they used amino acids at all. So a small subset of the 500 used by all life, bacteria, archaea, protists, in all organisms. Question? Well, life doesn't use them, but you can create them in the lab. They get created in natural environments. Yeah, question? And then, yeah?
Is it known whether the other forms of multicellular evolutions, like throughout history, use the same?
We're almost certain they did. I'll get to that question at the end. It's hard to answer with the data that I've given you so far. But it looks like these things were present in the last universal common ancestor of all life. That's why we all use it now, from a bacterium or not.
Are those 20 individual?
So let's do this. This is complicated. I'm going to leave it in. So what happens is Elliot and I exchange slides to see whether or not we think it's appropriate. So this is the genetic code. So I'm going to back up a step. Whoops. So it turns out that 3 base pairs code for an amino acid. And there's a specific sequence of letters that are code for each amino acid. And so as we go forward, this was worth a Nobel Prize. The first letter, the second letter, the third letter. And so each combination of letters codes for a specific amino acid. And then your protein is made up of, a typical protein has 300 amino acids in it. So it might be a leucine, isoleucine, leucine, threonine, proline, threonine, asparagine, proline, threonine, leucine, isoleucine. So a typical protein will have 300 of those. It could be 1,000. It could be 30 or 40. A prion, brain mad cow disease, is a little string of about 20 amino acids that get loose in the brain and then polymerizes and then starts to do serious damage. So it turns out the same combination of letters codes for exactly the same amino acid in all organisms, whether it's an archaea, a bacteria, a protist, a plant, a fungi, an animal. So again, if life had evolved independently in different times, you might expect, even if we all use DNA, and even if we all use the same subset of between amino acids, we would have had a different code for the different amino acids. But we don't. We have identically the same code. This is why it's called the universal genetic code. And then there's one or two little exceptions in some mitochondria. It's always little exceptions, like one or two slight differences. Some of these stop codons, which tells the cellular machinery to stop making the protein, is different. Last point. The machinery for making protein is highly complex and also universal. So here's a bacterium, and it's got these little green dots here that are things called ribosomes that make protein. Here is a picture of a ribosome. It is really complicated. That's the point. It weighs about the same as 3 million, well, that should be a +6, 3 million hydrogen atoms. So that's its weight. It's about 10 to the minus 8 of a meter. It's about a millionth of a centimeter. So take a centimeter, it's a millionth of that. And it's made up of an incredibly complex set of RNAs and proteins, and its basic structure is identical in all life. Bacteria, animals, plants, fungi, protists. Same genes. Okay. So those are just a subset of the complexities that all organisms share, which provide the strongest evidence that we're all actually derived from the single shared common ancestor. Now, last point is how can we compare organisms? Can we produce a universal measuring stick for all organisms? We have a huge range of morphological differences from worms to humans to trees to mushrooms to bacteria, which have virtually no morphology. So how are we going to compare them all? So I'm just going to introduce a term, disparity. Disparity is a term we use for measuring how different things look. So diversity is, say, the number of things, and the disparity is the difference between them. And so here I have a jellyfish, a sea urchin, and a rather large ant. This is a centimeter scale, so that's almost an inch. It's a bull ant, yeah. Look at the size of the jaws. I've never been bitten by one, but my cousins from New Zealand would come over and they would get bitten by them, and my brother and I were cruel and laughed. So in terms of their morphological differences, a jellyfish, a sea urchin, and an ant have vastly more differences than, say, 1,000 species of beetle that all kind of look like beetles. So disparity is measuring the differences, and diversity is the number of species. So bacteria and archaea have virtually no disparity in physical form. So there are actually two reasons why even the first protists, which were known by Linnaeus, were not included in the classification scheme, because the disparity was so low and the diversity was so low, it seemed inappropriate to create a brand new kingdom just for them. So they got sort of shoved into the gap somewhere between animals, plants, and fungi. So then, going back to these 6 major groups, animals, plants, and fungi have very high disparity. Bacteria, archaea, and protists have extraordinarily low disparity. So now I'm going to introduce the universal measuring stick of life, and it's called the small subunit ribosomal RNA molecule. I'm not going to ask you to memorize that. If it crops up in an exam, I will give you the name of it. There's no point in you memorizing that. It is part of the machinery that makes protein. All organisms have it. And it's very, very conserved. That basic machinery is so fundamental that evolution can't mess with it very much. So critically, we can recognize it in all organisms. So here's the ribosome again. This is the thing that takes the DNA. So the DNA with one set of machinery produces the single-strand RNA, and then that RNA is read by this, which creates protein. And so one half is called the small subunit, and the other half is called, we're not astronomers, so we don't call it the very large subunit. We just call it the large subunit. And then this blue here is an RNA. The white is another RNA, the pink is another RNA, and then all the rest of it is protein. So what one can do is one can sequence the nucleotides, the DNA or the RNA for this small subunit RNA, and fold it out on a page. So there it is there. This is from E. coli. And so it's hard to see where you're sitting, but here's a C, C, U, U, G, G, U. I can't see U, G, A, G, G. So that's just the string of nucleotides. And it's interesting, it forms a three-dimensional structure. Forms a three-dimensional structure that's critical to function. So DNA and RNA is not just information like in your computer. It also folds up into three-dimensional structures that have function themselves. That's pretty important. And then what one can do then is sequence, that is get this nucleotide sequence for the small subunit for all species. And then you can compare them. The more similar they are, the more closely related the species. So you can make a universal tree of life by comparing the RNA sequences of this particular molecule. And that's exactly what Carl Woese did at Illinois University that's in the Bryson reading. And it revolutionized our understanding of the relationship of organisms and the tree of life. And so then here is his tree, and it recognizes three great domains of life. I'm staggered by that, looks unreadable. And somehow or other, by just putting boxes around it, it looks quite simple. So there's the bacteria, one major branch, the archaebacteria, or the archaea, a second branch, and these are the eukaryotes, the third branch. And that's what he discovered. The archaea are completely separated from the bacteria. And so this was the first evidence that there were two great groups of bacteria, the true bacteria, eubacteria, and the archaea, the ancient bacteria, and then their names are reverted back to bacteria and archaea. So this is what Woese discovered, and these were completely separate from the eukaryotes, which are the protists, animals, plants, and fungi. It has been very difficult, and I'm not going to go into the detail here, but there's the root. That's where the last universal common ancestor appears to be, sitting that part on the tree. But we don't have any life to sort of determine what's the ancestor, because this is all life, the universal tree of life. So there's the split between eukaryotes and prokaryotes. Okay, so now, two surprises. Surprise number one, on this tree, Zea is corn, that's plants. That guy there is a fungus, a mushroom, and we have an animal in the house, Homo sapiens. So those are representatives of those three kingdoms, plants, animal, and fungi. Look at it. This tiny little tip on the universal tree of life. These three great groups that have been elevated to kingdoms that Linnaeus described, ignoring everything else. Well, he didn't ignore it, he couldn't see it. So surprise number one then is that, so let me just do this. So there's then plants, animal, fungi, and the protists. So surprise number one is huge molecular disparity in the protist, bacteria, and archaea. Absolutely phenomenal, dwarfing the things that we can see with our naked eye. This is not quite the same as the dark energy, dark matter, but a little bit. There's that which we can see, and then there's the rest of it, dark energy and dark matter. There's that which we can see, and then there's the rest of it that dominates the biological world that is invisible to our naked eye. Surprise number two, and this now gets to an earlier question that we had, which is, where do mitochondria and chloroplasts come from? So there's corn again, sitting there in the tree, and there's the corn mitochondria, and it has its own DNA, remember, and of those 35 genes, one of them is that small subunit RNA. So we can identify where the mitochondria goes, and there it is, sitting in amongst the bacteria, and I've colored it purple because it clusters with what's called the so-called purple bacteria. E. coli is a purple bacterium. So your mitochondria, the corn's mitochondria, is more closely related to E. coli in your stomach than it is to any of the cells in your body. Staggering discovery. Chloroplast DNA in the corn finishes up also being a bacterium. Its nearest living relatives are the cyanobacteria. But I showed you those green strands with the little round cell and the slightly squarer cells. Really, really interesting. So all the energy source that plants and animals use, ATP from the mitochondria and chloroplasts using sunlight, are actually derived from bacteria that are now sitting permanently inside our cells that make us eukaryotes. So this led to the theory of endosymbiosis. That is, that there's a symbiotic relationship between the eukaryotic cell and the mitochondria and the chloroplast. So the mitochondria are derived from an engulfed purple bacterium. The chloroplast are derived from an engulfed cyanobacterium. And this accounts for the reason that they have two membranes and their own DNA. Those few genes left inside are the remnants of whence they were once free-living bacteria in the free world before they got engulfed. And why the two membranes? So here now is that picture of an amoeba that I used as an example of the slime mold. So it has these sort of pseudopods where it creeps along and engulfs things. There's its nucleus. If it engulfed another cell, like a purple bacterium, but did not eat it, but this became a permanent resident reproducing inside the eukaryotic cell, then it would have two cell membranes, right? Its own and the membrane of the eukaryotic cell that engulfed it.
Do mitochondria and chloroplasts still live independently outside of our cells today?
Depends on what you mean. The nearest living relatives of the mitochondria are the purple bacteria, and so in that sense, their relatives are still out there in the real world.
But they've changed since then when they incorporated.
So they've changed radically when they got incorporated. So there are two bacteria that live in other organisms. Well, there are two bacteria that are almost identical to chloroplasts. One of them is called prochloron that lives inside a marine sponge that you find in Palau, and the other one lives in slime that was discovered in the Netherlands, which are almost identical.
So a follow-up question, can you take mitochondria and chloroplasts out of the cell?
Right.
And would they live?
So now let's push further then on how they radically changed. So one of the ways that they radically changed is the bacterium, the mitochondrial DNA consists of about 16,000 base pairs and 35 genes. E. coli has about 1 million base pairs, so 100 times larger, and has about 1,000 genes. So what's happened is it's very cozy living inside your own eukaryotic cell. Some of those genes got transported to the nucleus. Many of those genes simply got lost because the eukaryotic cell provides nutrient for the mitochondria to grow. So the mitochondria only have 35 left of the 1,000 genes that the free-living ones have. The chloroplast has 100 genes left of the roughly 1,000 that the cyanobacteria, or 2,000 that the cyanobacterium has. So no, they absolutely cannot live outside of the eukaryotic cell because they've given away almost all of their machinery that they need to live as independent organisms. So it's a very, very good question. Yeah? Are they what? Are they... No, they're made of the same stuff. Now, archaebacteria are made of something different, but the membranes are built of the same fats, lipids, like the membranes. They use the same proteins to make little channels in them so things can get in and out. The ATP, remember, is made in here and gets transported out across the two membranes. So no, in that sense, in some respects, we're just a eukaryotic cell is just a glorified, super large prokaryotic cell that engulfs something which became the nucleus, and then later engulfs something called the mitochondrion, with genes being transferred across, and then in plants later engulfs something called a chloroplast. The origin of the eukaryotic cell is a deep mystery. We don't have any eukaryotes that don't have mitochondria. But the nucleus is huge, it's got a double membrane, indicating that it engulfed something. So we don't understand the origin of the nucleus or the eukaryotic cell itself. We understand very well the origin of chloroplasts and mitochondria. My suspicion is that the chloroplast would die because we don't have the genes to recognize them and help them reproduce. I think that's what happen. Question.
Does the mitochondria reproduce with the cell?
Yes. So they reproduce with the cell. They reproduce inside the cell, but they get carried with the cell. And what's interesting about the mitochondria is that all of your mitochondria came from your mothers. None of it comes from the father. The sperm has lots of mitochondria, which it uses to swim, but only the DNA gets into the egg, and the mitochondria comes from the egg, so that you can trace your mother's lineage using mitochondrial DNA perfectly well. It's like a family name, except it's the mother's name, not the father's name, as in our patriarchal society. Question?
Just to clarify, prior to engulfment, the mitochondria only had one cell membrane, and they developed the second one when eligible.
Well, they didn't develop it. So they had first point, yes, when they were out in the free living world, they had one membrane. The second membrane is from the engulfing cell. Because if this engulfed, then there'd be a second membrane there. So this is what amoebas do. And then the second membrane is actually from the eukaryotic cell. So it's got its own membrane and then a membrane from the eukaryotic cell. Yeah, it's pretty wild. This idea is it's well over a century old now. There were some Russians, when they first started to do the microscopy to look at these things, noticed the two membranes. DNA wasn't known at that point. That was another 60 years in the future. So that was proposed. And then how many have heard of Lynn Margulis? Anybody? So she was the one who popularized it after taking a course on this subject at the University of Chicago, where she was an undergraduate. Question?
You said that archaeobacteria can derive energy from sugars. So do they have mitochondrial structures, or do they have mitochondrial to provide that?
So all organisms derive, bacteria, animals, plants, fungi, basically derive their energy from breaking sugar. Some archaea do it from other things like metals or hydrogen. They all use ATP. And so in some respects, all bacteria are mitochondria-like, in the sense that bacteria have a way of making ATP and then using the energy for it to do other cellular processes. Yeah.
So if the mitochondria didn't originally have two membranes,
Yeah.
why is it then, what DNA then allowed the cell to split and continue to give the mitochondria nutrients?
Yeah, when they reproduce, why do they produce with two membranes? That's a good question. I don't know how to answer that. I mean, I'm not sure how to answer that question. It's what happens. The nucleus has two membranes. It gets created that way too. So, the cell has lots of structures inside it that are all built from things. And there are instructions for building it, and the instructions includes the two membranes. So I don't know how else to answer that question.
So we don't know where those instructions, how those instructions get incorporated into the DNA.
I see what you're saying. Yeah, so right, that's a very good question, yeah. So this is something really important, which is a revolution that's ongoing and slow, which is there's a sense that everything is derived directly from the DNA and you can build it from the bottom up. In some respects, you can't build a computer that way. You can't give it a code and it will assemble the hard drive and the screen and the casing. So the casing comes from the previous casing, comes from the previous casing. The casing, the cell, comes from the previous casing, comes from the previous casing. So there's a lot of inheritance that goes from egg to egg to egg to egg that doesn't going through the DNA and back out to an egg again. So in some respects, that membrane is just because it's reproducing from one mitochondrion to the next without there being nothing in the DNA producing it from the beginning again, de novo. So it's really important. There's lots of inheritance from cell cytoplasm to cell cytoplasm that doesn't go through the DNA. So the mother's contribution is your legacy of your grandmothers and your great-grandmothers and all the way back down to things like this. And this is not well recognized, but beginning to be. Question?
It's a two-part question. First part, mitochondria burn out or die, and the second part are molecules recycle themselves.
Yes. So I won't put the slide up. I took it out. In every 2 weeks, approximately 90% of every part of your body, except the nerves and the bone, is completely rebuilt. Every 2 weeks, completely rebuilt. Everything is broken down and rebuilt over and over and over and over and over and over again. All your red blood cells, all your mitochondria, all your cells. So you are constantly being disassembled and reassembled. About 90% of you is replaced every 2 weeks. Obviously not your hair because that's dead. So yes, there's a constant... So we feel very static. We are not static. We're incredibly dynamic. So it turns out that there have been multiple endosymbiotic events of the photosynthetic machinery, not the mitochondria. So this is complicated. So there's the cyanobacteria at the top there. This is land plants there. This circle here represents the engulfing of whatever this thing was that engulfed a cyanobacterium to become a eukaryote with chloroplasts. So that's a eukaryote. It has a nucleus and mitochondria, and then it engulfed a cyanobacterium to become land plants. But each of these circles represents another one of those endosymbiotic events. So some things have gone through 2 or 3 of them. Evidence in part because some of them have 3 membranes, not 2 membranes. And so here's the brown algae, the kelp. So this is also part of the question. We said, why aren't kelps considered plants? Well, they have an additional endosymbiotic event, as well as being in a different part of the tree. Question? No, none of these are animals at all. They're all eukaryotes. They all have mitochondria and a nucleus. Most of them are single-celled, so they're protists. Well, they have mitochondria. So they must have fed on food. They get the cyanobacterium inside and said, hey, wait a second. I can just sit out in the sun. I don't have to worry about feeding anymore. So they went from having to feed off other organisms to saying, hey, I can just sit there and soak up the sunlight. So plants, that's a good question. So plants, before they did the engulfing thing, were more like animals in the sense that they were feeding off other nutrients, other organisms. And then when they engulfed the chloroplast, they said, hey, I can just sit there and soak up the sun. So in that sense, they have an origin similar to ours. That's a very good question. Yeah. Okay, so last slide, and it's a funny one. I was really struck. I was giving a lecture like this a number of years ago. Mitochondria. Go here, yes. So the surprise number 2 then, the energy, ATP and photosynthesis, that are the cells of... Wow, I can't read, I'm too close. The energy that cells use from multicellular organisms are derived from bacteria that are permanently reside in our cells. So that's the surprise. The energy that we use and depend upon is derived from bacteria that are really stripped down. They've thrown away most of their capacities, but are really stripped down and live inside our cells. So you might think rather differently about bacteria now. Right? If they weren't for them, we would absolutely not be here. So one of the great revolutions was this understanding. And so finally I was struck when I was lecturing about this. These things are mitochondria and chloroplasts, and I was watching a movie a number of years ago and struck by something that has a very similar terminology. The midi-chlorians, if you've heard of them from Star Wars. And I thought, you've got to be joking. Mitochondria, chloroplasts, midi-chlorians, mitochondria, chloroplasts. Mitochondria, chloroplasts, they live inside the cell, they harness the force. So clearly the writers of Star Wars learned about mitochondria and chloroplasts and were so taken by them that they decided that these would be the source of the force. So your mitochondria and chloroplasts are listening to the force. So that's it for this lecture. Next lecture, we're going to go back to astronomy and look at the fundamental force that dictates things, gravity. And so until then...